this third aspect is actually most critical, namely quantization of this vibrational energy, which is exactly what Debye did when he looked at lattices and vibrations of atoms and molecules in crystal lattices. Quantization, and the same thing that Planck did for electromagnetic radiation, introducing a formula that energy is proportional to frequency through a constant, but the frequencies and wavelengths are not continuous, they are discrete. Namely, there are boundary conditions, and you need to introduce boundaries. Membranes are clear boundaries in living systems, and that leads to this argument for quantization. This is a complicated metabolic pathway composed of three major steps: glycolysis, citric acid cycle, and oxidative phosphorylation. This, in the last step, in the oxidative phosphorylation step, we had the respiratory chain reaction where eventually, in a cyclical manner, you produce ATP from the addition of a phosphate group to ADP, and that is coupled to the electron transfer chain operation, which is coupled to proton transfer. And remember, we talked about the proton transfer also emitting electromagnetic radiation in the signaling and interaction between cells. So all of this together makes a lot of sense in various contexts, but first of all, from the point of view of quantum metabolism, most of the quantum processes take place in the inner mitochondrial membrane and through these 5 protein complexes. Without going into details, this cyclic operation of low energy electrons being transported to high energy electrons and back down in a cyclical manner is involved in the electron transport chain operations, and of course depends on ATP concentrations, ADP concentrations, a lot of details going into it, but the main thing is, if we assume that this is a cyclical process, is number one, and then the process of these taking place in mitochondria by a number of FTPases is coupled not only in a given cell, but across cells in an organ and across organs into an entire organism. If we are synchronized properly, our metabolism is coordinated in the entire body. Not simply one organ doing its own thing and the other organs decoupled from it. It doesn't happen in normal physiological conditions. In pathology, perhaps, as actually has been argued that, for example, in the case of cancer, this decoupling occurs. Cancer cells decouple metabolically from the surrounding normal cells, actually move from oxidative phosphorylation mode of energy production to glycolysis. The last quantum steps do not happen, or at least happen at a much lower rate. And that the more decoupled cancer cells are, the more malignant the disease becomes. So the quantum synchronization or quantum metabolism is a hallmark of a healthy metabolism, whereas gradual transition to classical decoupling between mitochondria and actually even shutting down of mitochondria is a hallmark of pathology, and in fact, in the end, apoptosis. Apoptosis is characterized by the shutting down of mitochondria. Okay, so these are the three conceptual components that Demetrius combined. Chemiosmotic coupling proposed by Mitchell, Nobel Prize there. Energy storage in the form of high frequency oscillation, and about the same time actually. Probably didn't get a Nobel Prize. He should have, because he was one of the early physicists who explained superconductivity. And then, actually there's a Froley Hamiltonian, which is underlying the theory of superconductivity. And finally, energy quantization, which is I wouldn't say mimicking, but taking inspiration from the Debye in crystals and couples these metabolic oscillators, just like atomic vibrations move nodes in a crystal lattice back and forth at a given frequency. The same is true for metabolic oscillators. They're coupled with a frequency. These frequencies may change from system to system, but they are coupled, and the coupling is critical to the establishment of quantization. So these are the assumptions. Enzymes, metabolic enzymes are embedded in a network spanning the entire organism. They undergo electrochemical oscillations. These oscillations involve transfer of electrons, so they are quantum mechanical par excellence, and these oscillations are quantized. So this is the assumption, very much like the Planck quantization. This is applied to metabolic oscillators, and you can find, this is the original reference to Demetrius in 2003, where he proposed Planck-like constant in the context of metabolic enzyme oscillations. And then the rest is a little bit of mathematics, not too heavy. It has been done almost a century ago for the first time, and that means you do this. You calculate the averages. The difference between physics, calculation physics for the specific heat of solids, a la Debye, and for metabolic oscillators is that we are looking at not the equilibrium energy, but the power or energy over time, metabolic rate, power generation rate, and that is corrected for in the steps of this calculation. So these are the ingredients going into the theory. There's a maximum frequency of these oscillators, and then one obtains the energy as a function of characteristic cycle time, and then there's a scaling of the cycle time with empirical, with the size of the organism, with the 1/4 power that I showed earlier, and the end result is this. But basic metabolic or basal metabolic rate P scales with the mass or the weight of the organism to the power d over d plus 1. Why is that? Well, d is spatial dimensionality. I mentioned to you that in crystals too, depending if it's a one-dimensional line of atoms, whether it's a two-dimensional plane of atoms or a cube of atoms, you have different formulas for the specific heat of solids. For organisms, the basal metabolic rate also ends up being dependent on dimensionality. If you can imagine a very elongated organism, almost one-dimensional, maybe some plants can be seen this way. In the space of biological systems, actually, you can even think of fractal dimensions, but easily planar leaves, planar structures, or more complicated geometries. But in the simplest case of linear, two-dimensional, flat, if you will, or bulky, you'll have different scaling laws. In the case of one-dimensional linear objects, you'll have 1/2 power law, which we saw as being observed in numerous species. When these two, for flat, like leaves and plants typically, because they absorb light into a plane, the power law is 2 divided by 2 plus 1, which is 2/3, which is one of those almost canonical values. And then if these three, you have 3 divided by 3 plus 1, which is 3/4, which is the most common. So most of the biological organisms would be considered bulky or three-dimensional. And in this case, you obtain an explanation of this 3/4 power law, which is the most dominant, by this kind of calculation. And there is one more. When d goes to infinity, you get 1. What would that be? I guess that the concept of dimensionality in the case of biological systems would be perhaps how many neighboring oscillators one enzymatic oscillator has. So if you can think about one-dimensional situation, you have two, one on the right, one on the left. In two-dimensional, you will have in front and behind, left, right, So you have 4. If you have, so 2, 4, of course 6 in 3 dimensions. If you have a huge number in all directions, you can kind of approximate it by higher and higher dimension. So it's basically this dimensionality has to do with the number of nearest neighbor oscillators. That's my interpretation. I'll skip this because it's kind of an interesting comparison between solids and cells. In solids, temperature is the knob you can change and observe the response of the system, the crystal in this case. In living systems, actually, we don't touch that knob because that knob is dangerous. It will kill the system either way if you turn it up too high or too low. But what we can do is change time, slow things down, slow down the delivery of nutrients or oversupply. So that is the knob that is controlling the processes. Heat capacity in solids, metabolic rate in biological systems, cells, and instead of thermodynamic entropy, which is a byproduct of energy supply entropy, in living systems, entropy production rate or heat dissipation. So this is interesting, and I told you that one of the assumptions that Demetrius made in the beginning of one of the key assumptions is the quantization of these oscillator frequencies. And so the question might arise, what is the comparable biological Planck constant? And I think this is a busy slide, but I'll just give you the gist of it. You can come up with a number, actually, that links energy in the context of biological energy production or metabolic energy with the frequency of ATPase, for example, turnover. And so that goes, this is the energy production in the human, 10 to the 21 ATP molecules per second. That's the rate at which is a blindingly fast rate of ATP production, 10 to the 21st power of ATP molecules per second. And then there is the number of cells in the human body, which is 35 trillion, roughly. And then each cell has about 1,000 mitochondria, and each mitochondrion produces about 30,000 ATP events per second, and then each ATP event has a millisecond time scale. So to cut to the chase, you compare the energy to the frequency. Frequency is inverse time, so millisecond gives you 1 kilohertz, and you obtain K from all these numbers, and it turns out that this, or kappa, this biological Planck constant obtained by putting in the numbers known from human physiology gives you something like 10 to the minus 24th joules times second. The actual physical Planck constant is 6.6 times 10 to the minus 34th, which means that the ratio between biological and physical time constant is something on the order of 10 to the 11th, or 100 billion. Biological would be 100 billion times bigger, or 200 billion times bigger than physical. Why is that? Actually, it makes a little bit of sense, because a physical Planck constant comes from a single elementary particle. Right? Electron is the particle that is subject to the Heisenberg uncertainty principle, we have the Planck constant, or the Schrödinger equation, we have the Planck constant, and so on, for a single particle. But how many, so the question is how many particles are there in the sphere of influence of a single mitochondrion and so on, of which you have 10,000. So I did the calculation. It gives you something like 200 billion atoms in that sphere. So it's exactly that number. In other words, it's a synchronized, a synchronized area, so to speak, energetically or energetically affected, which is so much bigger than a single atom or a single particle involved in quantization processes in physics. So there is a bit of rationale for this number. It's not completely out of the blue skies. It will tell you that these processes, which are non-equilibrium, affect a large group of atoms, and therefore they are much slower. Atomic processes will be on a femtosecond, 10 to the minus 15 second in physics. In biology, these molecular cycles are milliseconds, 10 to the minus third. So again, 10 to the 12th is more or less the ratio, slower and larger. The bigger this side of a system, the slower the process. Okay, now let's move on to evidence for quantum coherence elsewhere. And I mentioned photosynthesis, so far the best studied biological effect which has quantum physics involved directly in its operations. And that the story, it goes back to 2007, where Engel and collaborators provided experimental evidence for quantum beating in chloroplasts, and that followed with other researchers showing electronic coherence in photosynthetic complexes of different types, including also theoretical work showing a model that gives long range and multi-particle, relatively long time quantum coherence in these systems. So let's start with photosynthesis. And I guess this is really in a logical sequence because photosynthesis is a metabolic process for plants and some bacteria. So this is how energy is produced in plants from photosynthesis. We don't do this, as far as I know, as humans or mammals. We consume nutrients like glucose, sugar, and then produce ATP out of that. By photosynthesis, the end product is the same, but the input is different. Photosynthesis uses the energy of electromagnetic radiation, visible light, with the presence of water and CO2, and generates electronic energy flow to reaction centers where ATP molecules are synthesized. What's interesting is the chromophore, where this quantum processing takes place. The molecules forming it are spaced on the order of 15 angstroms. 15 angstroms to 20 angstroms, more or less, to our knowledge, the maximum range over which quantum tunneling can occur. So plants operate within the allowed range of quantum tunneling. So that's number one. And are very intricately composed using light harvesting complexes, those that can be photoexcited, if you will. And this energy is transferred to photosynthetic reaction centers where the energy is collected and transformed or transduced. In the end, chemical energy is obtained in the form of ATP molecules, the biological energy quantum. So electrons turn CO2 into organic compounds. And this has been studied, especially in the so-called FMO, Fenner-Matthews-Olson complex in some bacteria, for example, C. tapetum, found in New Zealand, in some hot springs. But when you look closely, you find some very symmetric structures in this FMO complex. Light is captured and is transformed through a number of steps, activating various molecules, but the most important one actually is shown here in the next slide. The FMO complex is composed of 7 rings, 7 multi-cyclic rings, shown here, and they are spaced, as I mentioned, within the 1.5 angstrom distance, so they can actually tunnel quantum energy, or share, in fact. No matter which one of these you absorbs light, all of them will share the energy. Electrons will be activated in all. And this quantum excitation can be modeled, and calculations as well as experiments demonstrated that it can be supported for nanoseconds, which is long enough for the chemical reactions to take place in the system. More recently, single-celled algae have been demonstrated to be performing this light harvesting process using quantum coherence. And the commonality is that these light harvesting complexes have a very similar structure, and there were some very detailed experiments demonstrated quantum entanglement, and in the laboratory setting. This experiment by Engel showed the presence of quantum beats. So this involves three beams of light impinging on the quantum mechanical system, and this the light harvesting complex, and the emitted radiation is detected and shows to have the characteristics of quantum beats. This is a more detailed illustration of the system. The sample is here, and the laser light is split and recombined at particular time separations, giving rise to quantum beats at the sample, and then analyzed in the spectrometer. This is experimental data showing you the coherence wavelength, so the wavelength of the light that is actually absorbed and coherently shared within the complex, and that's in the range of around 800 nanometers. So it's near visible. And you have these characteristic quantum beats over, in this case, 660 femtoseconds, but they can be longer. So that's a quantum superposition of states taking place in this 7-ring complex. So not only a single electron can be in 2 states, but here the whole complex can share a wave function that is in 2 states, superposition of 2 states. You can look at it as a search algorithm, because I mentioned earlier that quantum systems can explore the entire space. The state is delocalized. It's not sitting in one place and then hopping to another. That can happen too, but in this case, the state is distributed over the entire complex. So it's searching for the lowest energy state in which to which it can funnel or travel. So now we move on. So that's basically the best documented example of quantum states in biology. Whether it's quantum biology or quantum physics in biology, that's a delicate question. It is definitely quantum physics in biology, but the issue is whether biology has its own quantization rules, like Demetrius proposes. It's not, in the case of metabolism, it's a bit different. As you remember, I mentioned this biological quantum Planck constant, which is different from the physical. here we operate in photosynthesis with standard physics. We don't have to bend it or change it, it just apply. I mentioned olfaction earlier. There are sort of competing theories of olfaction, whether the sense of smell is operating just like other receptors on the principle of lock and key, or i.e. shape matching, or something else. And shape matching actually is not easy to explain many of the observations, empirical observations regarding the sense of smell in humans and animals, because receptors, odor receptors, olfactory receptors, recognize molecules which have no similarity in shape, but they may have the same vibrational or vibrating quantum states. So that's the Lucas-Turin theory of olfaction, which has gained some traction in the community. But I think the jury is out. There's still a lot of controversy and discussion about it. If it's not true, then we don't have an explanation that is general enough. In some cases, yes, but not in all. Now, back for a few minutes to photosynthesis like exciton generation. So it's quantum physics in biology. And recall I talked about Albert Buller's experiments where centrioles were hypothesized to absorb electromagnetic radiation in the near infrared range and respond to it. So microtubules were involved in this. What's interesting is microtubules are so multifunctional. Microtubules are shown here performing one function, namely cell division and dividing cells. They are shown here and here, connecting centrosomes to chromosomes and segregating chromosomes and dividing cells. So this is one of the key functions, but it has microtubules have more, including transport. Motor proteins such as kinesin, dynein move in microtubules. Microtubules also provide structural, mechanical support to many cells because they are very rigid. And finally, they are implicated in neuronal processing, and we'll come to it maybe on Thursday, not today. But I want to say something about microtubules in connection with photosynthesis. Because when you look at the structure of microtubules, you see great regularity or symmetry of the lattice of tubulin. Tubulin is the building block, the protein that makes microtubules. The stable form is beta alpha tubulin dimer, and it also has aromatic tryptophan molecules with the indole rings, which are very similar in structure to those multi-ring complexes, light harvesting complexes in photosynthesis, as you will see. And the distances between these groups, tryptophans, actually are comparable. There's 8 tryptophans per tubulin dimer, and there's 13 dimers in a ring forming a microtubule. So each dimer is about 8 nanometers by 4 nanometers by 5 nanometers. And therefore it makes sense to analyze it in the context of electronic degrees of freedom that can be excited by electromagnetic waves. This is the architecture of microtubules in neurons, the axon and dendrites, and they are forming parallel bundles, which would be conducive to signal propagation, for example. If this was an electronic signal, it could propagate in parallel pathways and down the axon into dendrites, and etc. Here is another representation of these pathways that excitons can traverse in a patch of tubulin forming a microtubule. Microtubule is cylindrical, but the surface is hexagonal symmetric. This is a single dimer of tubulin, which is the building block of microtubules. And I'm showing here a very clearly visible structure of a tryptophan neighborhood in one dimer. And you have 2, 4, 6, 8 of them. Their geometry is very similar to the geometry of the ring structures in light harvesting complexes, and the distances are also similar. And together with my former students, Travis Craddock and Stuart Hammerov, we've developed a quantum model of exciton energy absorption and transfer in microtubules based on the transition dipole moments in tryptophans. The transition dipole moment is about 5.5 dBi. The interaction is dipole-dipole with a given strength, and tryptophans can be excited in the 260 to 305 nanometer wavelength range, which is ultraviolet. And this excitation has been simulated by computational work and published by us in collaboration with Travis Craddock, as I mentioned. And we demonstrated that very much like in the photosynthetic systems, you can create these collective excitations that propagate. And first of all, they are shared within a dipole, and they can propagate along the microtubule. I will finish this class with something that adds credence to it by recent experiments which are not published yet. So you are the first audience to see it. These experiments deal with the demonstration of microtubule capability to absorb electromagnetic radiation and store it over a long period of time, which would be incredibly interesting for many reasons, one of which would be to explain the antenna-like property of microtubules in centrosomes, for example, that Albert Buehler talked about when he talked about cell intelligence, cells talking to other cells via burst of electromagnetic energy, and you need to have an antenna. Microtubules in the centrosomes could be that antenna if we prove that they can actually absorb and keep it for long enough, not scatter, not just dampen it, but keep it and maybe re-emit. So this leads to the topic of biophotons. Again, we are looping back to Gurevich, Alexander Gurevich, the Soviet, Russian first and Soviet scientist who proposed the existence of morphogenic fields, biophotons, perhaps. And there is a lot of scattered information about the existence of biophotons. Biophotons means not, as I said again, infrared emission, but photons which are emitted by living systems outside of the normal thermal theory. They are emitted at the non-equilibrium rate, something outside of the thermal prediction. And there is a lot of information that is scattered, not really very well organized. There's a nice review paper from the group of Michal Cifra in Prague, in the Czech Institute of Photonics, published a couple of years ago, which reviews the entire literature on biophotons. And people have measured the intensity of photon emission from living systems, including cells, cancer cells, normal cells, human body overall, skin, organs, brain, kidney, plants, leaves, onion roots, and so on. And there is evidence that these are very weak amounts. Typically, that would be something like between a few to maybe 100 or a few hundred photons per centimeter squared per second. So low intensity, very low intensity, nonetheless measurable and present. That's the range. And so just to keep in mind. This is, of course, orders of magnitude lower than light that we are exposed to from the sun. But some phosphorescent organisms emit photons at a much higher intensity, for example, fireflies and luminous bacteria. But normally you would be in this range. Okay? So the experiments I will tell you about were performed by Professor Aristide Dogariou at the University of Central Florida in Orlando, and he used photons of a particular wavelength from laser and exposed microtubules in a sample, as well as tubulin and the buffer itself, and measured the re-emitted photons. In other words, how much of these photons would be emitted after a while. And this is called delayed luminescence, because normally you wouldn't expect any delay, or very little, maybe picoseconds. This is the transition to an excited state and re-emission, and that's it, gone. Well, delayed luminescence is also a technical term, and there's a lot of biological, again, scattered literature that some biological systems exhibit this delayed luminescence. And one of the examples is phosphorescence, which also applies to chemical molecules that can be artificially constructed to exhibit phosphorescence. They store electromagnetic radiation energy and re-emit over seconds to minutes, even hours. And that is something mind-boggling when you think about quantum states. We talk about femtosecond transitions at the quantum level, and suddenly you have quanta which are trapped or frozen for minutes, many minutes, and then released. So a long-lived quantum state, in a way, a dream come true if you want to use quantum mechanics to explain, for example, our consciousness or the way the brain works. If we could trap an electromagnetic energy, use it, and then release it. So this experiment was done with a illumination of the sample over 2 minutes, and then 4 minutes measurement of the released radiation, and then repetition a number of times. This is the setup with the technical parameters, and the wavelength used in this particular case was 630 nanometers, which is about 2 electron volts in electron volt units. So that's visible, and okay, that's about it. Low intensity, 0.061 milliwatts per millimeter squared, which would correspond to those numbers I gave you in living systems. It's not really flooding the system with light. It's very low intensity and uniformly applied from all directions. So this shows you the results, which I want to talk about for a few minutes. Tubulin, unassembled in microtubules, shows a few times lower intensity of re-emitted radiation. It still has surprisingly long decay time, which is maybe a minute or two. The microtubules in random orientations, much higher intensity and somewhat longer re-emission time, and eventually, in the last case, microtubules oriented by the field, and you have much higher yet, the delayed luminescence profile, which is actually intriguing because we are talking about minutes, 5, 6, half lifetime on this would be from 16 to 8, let's say in this case 6 minutes. In other words, you shine laser light on a microtubule or a bunch of microtubules in solution, and then you measure how long this light is re-emitted, and it takes 6 minutes, 5, 6 minutes to re-emit. It's amazing. That means there is a quantum state created in the structure over a long distance and over a long period of time. There's new data I'm not going to show, which indicates actually the length dependence of this delayed luminescent state. So we're getting closer. We're at zeroing in on quantum, long-lived quantum states and biological structures. This is not a living system, but it's a very important part of the living system. So, yeah. To close the loop, we started with Gurevich morphogenic fields. Now we're coming back to this at the level of understanding the mechanism, perhaps. Microtubules are ideal structures which could absorb and re-emit, and maybe transduce electromagnetic quanta through electronic degrees of freedom. Mitochondria are ideal structures, so again, back to metabolism, that produce electromagnetic radiation as they act, function by producing ATP, and the byproduct is emission of photons. If mitochondria stop for some reason, damage or cancerous transformation, this is gone. Then you suddenly are not communicating with the neighbors. You're not saying, okay, I'm alive, I'm producing metabolic energy. And interestingly, and not everybody knows, but cell biologists know, mitochondria move on microtubules using kinesin motors as transport vehicles. So all of this is interconnected. Mitochondria are very closely associated in in cells which are not in a state of mitosis, because in mitosis they separate. But in interphase, mitochondria move around the cell on microtubules, and that can be a closely linked communication system. Emission, absorption, re-emission. I will stop at this point. I think we've covered a big chunk of what I would call quantum biology or examples thereof. And on Thursday I'll talk about things which are a bit far into the field of more speculative quantum biology, maybe quantum medicine too. Thank you.